Hi everyone, my name is Huma and my research was based on investigating the effect of sulforaphane on melanoma cells viability, proliferation, metastasis, and hTERT gene expression. Sulforaphane is a compound which is found in cruciferous vegetables like broccoli, sprouts, etc. This compound has shown potential to reduce many different types of cancers. The way it does this is through various apoptotic and epigenetic regulations. This study primarily focused on epigenetic regulation of SFN on telomeres of chromosome of cancer cells. Telomeres are the protective end of chromosome that naturally shorten during cell divisions. This is because of decreased function of human telomerase reverse transcriptase gene, also called as hTERT gene. These genes are responsible for maintaining the length of telomere. However, in cancer cells, this gene is excessively expressed, preventing telomere from shortening and causing continuous cell division. hTERT accomplishes this by altering the methylation status of DNA and acetylation status of histones, which results in decreased proliferation since the function of hTERT is hindered. This takes us back to our research objective, which was to investigate SFN anti-proliferative effect, decreased viability and metastasis, and downregulation of hTERT gene. These were all accomplished by conducting different types of assays such as MTT assay, RT-qPCR, clonogenic assay, and wound healing assay. So for all the assays, the cells were first cultured in DMEM medium with 10% FPS, 2 millimolar L-glutamine, 1% penstrep at 37 degrees Celsius and 5% CO2. For all the assays, the SFN treatment were prepared by dissolving the SFN capsules in PBS, which was sonicated for 20 minutes at 37 degrees Celsius and then centrifuged to collect the supernatant. The concentration of the SFN in the extract was determined by running it through UPLC against a standard curve of SFN. Once the concentration was determined in the extract, the assays were performed. So for MTT assay, the cells were seeded in 96 well plate, which was then treated with SFN along with vehicle control. Following the treatments, MTT was added, which was incubated for 2 hours, and then DMSO was added to dissolve the foams and crystals formed by the available cells. The color change was absorbed by reading the absorbance on the plate reader and was analyzed. For RT-qPCR, the cells were first seeded and they were treated with SFN along with vehicle control. Following this, RNA was extracted, cDNA was synthesized, and PCR was conducted for hTERT gene and GATA gene, which was the normalizing gene. The clonogenic assay also looked at the proliferation ability, which was performed by seeding the cells at very low cell density and then treating with SFN. Following treatments, the colonies formed were stained, fixed, and then counted. Lastly, for scratch assay, the cells were seeded again and wound was formed on the wells of the plate. The wounds were then treated with SFN along with vehicle control, and the wound was observed after 24 hours. The result for the MTT assay showed a reduction in cell viability and proliferation significantly for the doses equal to or greater than 20 micromolar. And one of the reasons for this could be reduction in hTERT expression, which we note from literature. This was proved by RT-qPCR results, which showed significant downregulation in hTERT gene when treated with 20 micromolar of SFN. The clonogenic assay also showed no formation of colony in SFN treatment cell. However, there were colonies in vehicle control. This suggests that the reduction of hTERT expression caused a decrease in ability to form colonies and proliferate. The scratch assay also showed a significant decrease in wound closure post-SFN treatment, indicating reduced metastasis behavior. Overall, this study showed promising effect of SFN in reducing viability, proliferation, metastasis in melanoma cells, along with downregulation of hTERT gene. Therefore, for further investigation, more doses of SFN can be explored, which will help to study their impact better. And for deeply understanding the epigenetic alteration, other assays can be performed like telomere activity assay, histone deacetylase assay, DNMT assay, and bisulfite sequencing assay to measure the DNA methylation status. Also, it would be interesting to explore the apoptotic pathways concurrently. Additionally, studying its effect on non-cancerous cell line can also be useful. Furthermore, exploring its impact with other cancer drugs could uncover some synergetic effects that it would have. Finally, I would like to thank my professors Michael Gibson and Jessica Livingston and all the lab technicians for helping me throughout the conduct of this research.